Hello, my name is Skylar Kosegi, and today is January 6th, 2024, and I'm doing my presentation on the anatomy and enzymology of fat absorption. Okay, so the main enzyme that's involved in this process is lipase, and there's three types in our bodies. There's lingual, gastric, and pancreatic lipases, which are secreted by those respective parts of the body. And its main action is to break a triglyceride into a monoglyceride and two fatty acids. So you can see on the diagram to the right how the enzyme works. So it's going to bind to the substrate, which is the triglyceride. It'll form an enzyme substrate complex, which will facilitate the breakdown or the hydrolysis reaction to break it into smaller parts, and then the product is released. The first place where lipids can be digested is the oral cavity, but there's actually very little fat absorption here. It's just starting the process of breaking down the triglycerides. So here, it's usually just breaking a triglyceride and taking off one of the fatty acids, leaving a diglyceride left, which can be passed down in the digestive system. And then lingual lipase is secreted by the von Ebner's gland into the saliva, which you can see to the diagram to the left. And then moving on to the stomach and gastric lipase. This is secreted by chief cells of the glands in the stomach, and you can see what that would look like on the diagram to the right. And then this is beginning the breakdown of short and medium length chains. And again, it's a hydrolysis reaction because you have a larger molecule and you're breaking it into smaller parts. And the enzyme is released inactivated, and it's activated by the acidic environment of the stomach, so it has an acidic optimal pH. And it still has limited activity in comparison to the pancreatic lipase, but it's just still continuing that process. So then moving into the small intestine, the fats or the lipids will come into contact with pancreatic lipase, and this is released in the small intestine. This makes it in the pancreas, and it's released in the small intestine as a response to presence of lipids in the chyme. And this usually acts on longer chains of triglycerides, and it's the part that's breaking them down for the most part into two fatty acids and a monoglyceride, which you can see below. And then those are the two products that can actually be absorbed by the small intestine. And then another thing to mention about the digestion of lipids in the small intestine are bile salts. So when you have lipids which are hydrophobic in a watery environment, they tend to clump together, but this has a very low surface area, and it makes it hard for the lipases to actually react with the to catalyze reactions with the individual triglycerides. So bile salts are made in the liver. They're a part of the bile, which is stored in the gallbladder, and it's released into the small intestine through bile ducts. And their job is to emulsify those lipids that you can see are like clumped together, and they'll be kind of like separated into their individual parts where pancreatic lipase can actually catalyze its reaction. So it'll increase the efficiency of that enzyme. And then these are stimulated by CCK, and that's released when the body detects that fats are present in the small intestine. So then fats are actually absorbed for the most part in the small intestine. Those monoglycerides and the individual fatty acids that have been breaking down since they entered the oral cavity, they're passively transported into the enterocytes of the small intestine, which you can see to the right. And then when they enter the enterocytes, they're actually reassembled into triglycerides again. And then they enter the Golgi apparatus where they're packaged with other proteins, and these form chylomicrons. And then these exit the enterocytes via exocytosis. They enter the interstitial space, and then they actually are entering the lacteals, which lead to the thoracic duct, and they enter the lymphatic system. And then this is my student activity I made. I think active recall is important once you watch a lecture to not just reread your notes, but also quiz yourself on the material because it helps it stick longer. So these are just some basic questions that students can ask themselves after the presentation to check that they are starting to memorize things and understand the process as a whole. But thank you.